Hello, my name is Jillian Collins, and I'm a second year PhD student in Dr. Farnsworth's lab. And we are a fairly new lab. We currently have 3 grad students and 3 undergrad students. And so our work is primarily focused in type 1 diabetes. So we're a pretty big bio lab, but we also do a lot of work with biomaterials. So if you are also interested in biomaterials and not just straight biology, you would definitely fit into this lab as well. So to start off with, what is type 1 diabetes? And it's essentially an autoimmune disease that can be characterized as the immune-mediated killing of your beta cells, which are responsible for secreting insulin and maintaining your glucose homeostasis. So the consequences of this disease is that your immune system sees insulin as a foreign invader. So it then goes to whatever is producing insulin, which in this case is your beta cells, and it kills them. And the problem with this is without insulin, you cannot regulate your glucose levels and you become what's called hyperglycemic, which means you have too much glucose in your bloodstream, not being used for energy, and it results in all of these major complications, kidney failure, heart failure, etc. And you will eventually die if you can't get it properly regulated. And our research specifically is working with this protein called protein kinase C delta. So we're kind of looking at more of the downstream stuff of the immune response. So what we found so far is that there are these things called proinflammatory cytokines. So we have found that these are the little guys, they come and attach to the beta cells to initiate what's called apoptosis, which is natural cell death. So there's 2 types, there's necrosis and apoptosis. Necrosis is where something physically comes in and is killing the cell. Apoptosis is where the cell just either dies on its own or something attaches and is sending signals to the cell saying, hey, you need to start initiating this process, like you're not a good cell, like you basically need to die. And so we're focusing on the apoptotic process. And so what has been found in cancer cells, for example, is this protein kinase C delta. It plays a major role in regulating the apoptotic process. So what they found is that when the cytokines come in, it'll bind to the outside of the cell and it will initiate PKC delta, which means it typically resides in the cytosol, and it will then translocate from the cytosol to the nucleus, where it's then cleaved by this other protein called caspase 3. And that cleavage is what activates the catalytic fragment of PKC delta, which continues the stages of apoptosis. So we were thinking that there might be a similar mechanism within beta cells. So we hypothesized that this same protein, PKC delta, goes through a very similar mechanisms within the beta cell. So how we tested this was, if we knock out PKC delta, does that show a decrease in beta cell death? So we did some genetic engineering with this particular mouse model where we reduced PKC delta activity by 58%. And what we found, as you can see in the results section, that by inhibiting PKC delta, it actually protected both mouse and human islets. And an islet is where the beta cells live. So if the islets are alive, then you have like happy beta cells in there. So again, what we found is that in both mouse and human islets, you can see in the presence of cytokines, which is the initiator of the apoptotic process, you see less apoptotic cell death. So we were like, sweet, okay, we know that knocking out the expression or inhibiting PKC delta is working. So our next step was, is PKC delta actually translocating from the cytosol to the nucleus? So uncovering this mechanism. And what we found, as you can see in the first figure, figure A, is that there is much more green in the 24 hours with the cytokines, which is showing you that there's a lot more PKC delta activity in the nucleus, which is supporting the conclusion that yes, PKC delta is not only translocating, but it's also being activated within the nucleus. And you can also see that with the figure B and D, with the nuclear to cytosolic ratio, you see a much higher activity of PKC delta after cytokine stimulation in both. So now step 2 is completed. We found that inhibiting it not only protects beta cells, but PKC delta does in fact translocate from the cytosol to the nucleus. So finally, the final mystery is cleavage via caspase 3 actually required for PKC delta to initiate the final stages? And we found that in fact, it is required. If you look in that figure up in the top right, you see that there is a thing that says CMGFP PKC delta, which is the blue bar. So that is a cleaved mutant, which means that caspase 3 cannot cleave this PKC delta. And as you can see, if caspase 3 cannot cleave it, you see less percentage of the apoptotic cells within the islets. So putting this all together in our conclusions, you can see that when the cytokines bind, it does stimulate PKC delta to translocate from the cytosol to the nucleus, where it is then cleaved by this caspase 3, and then it undergoes the remaining process of beta cell apoptosis. So moving on, what are kind of our next steps, which is what I'm doing, which is, okay, well, what if we can deliver a drug that consistently inhibits this protein? What would that do over a 1 to 2 month period? So this is where the biomaterials comes in. So I will be basically fabricating nanoparticles with a drug that we have, and we're going to try to deliver it in mice and see basically, does that actually help prevent type 1 diabetes in this mouse model? So that's just kind of the basics of what I do. The other projects offered in our lab they are trying to make a biomimetic scaffold. And what they're trying to do is essentially simulate type 1 diabetes outside of a mouse or a human. So it's like, you don't have to wait for a mouse to either naturally progress into the disease. You don't have to wait for human samples. You don't have to inject mice to make them diabetic. So that is currently our other projects. And you make a lot of polymer synthesis. So you get to do a lot of work with NMR, SCM, learning how to use a lyophilizer, et cetera. You definitely learn a lot of skills, which is awesome. And one of the other neat things about our lab is we do our own animal work. So not only will you learn how to handle animals, but you'll also learn how to conduct and plan experiments with them, which will be a huge advantage if you want to do anything in the biofield, whether it is straight biology or biomaterials. And we do this at this place called Anschutz, which is that building in the top right-hand screen. We do a lot of collaborative work with them. So that is where we go to do all of our animal work. And what's awesome about this place is we get to attend a lot of different seminars. We get to attend, they're called DRC seminars, which happen weekly. We get to attend the islet supergroup meetings, which happens, I believe, the third week of every month. And there's all these other seminars that are offered that you can go see if you're really interested. And if COVID wasn't a thing, you got to actually meet the speakers after these talks. So you get to meet all of these people from around the world. And they also have other equipment that we used to use. They have a confocal that we would use, but because we actually just got one, you no longer have to go there for imaging, which is super nice. And I mean, I don't know, our group is a lot of fun. And if you're really into biology or biomaterials, this is a great lab to join. We're definitely small and close-knit. And yeah, that's kind of all I got. So thank you for listening, and yeah, have a good day.